Hello, I'm Dr. David O'Connor, and I'll be talking to you today about monitoring and modelling pollen. Before I start, I just want to thank all the people who are named here. Okay, without them, this work wouldn't be possible. I'd also like to thank the EPA and Met for funding this work. Of course, the first question I generally get asked about why I monitor pollen is why do we care? And the most obvious thing to talk about is, its effect on our well-being. We all know someone who suffers from hay fever. Okay, and it can be an awful thing having those kind of flu-like symptoms in the middle of the summer. Okay, and about 20% of adults within Europe suffer from some form of allergy. Another reason why we're interested in pollen is they can affect the climate. Okay, pollen and other bioaerosols can cause cloud condensation nuclei to form clouds. This can affect the radiative balance of the Earth. Okay, and that's very, very important when we want to model and forecast climate into the future. Climate can also affect the concentrations of the pollen in the atmosphere and also how long the pollen season will last for. But the big thing I want to talk about is its effect on health. Okay, yeah, it's nasty if you have flu-like symptoms in the middle of the summer, but people who have underlying conditions like asthma can have a huge detrimental effect on their health. It can trigger asthma attacks. So in a sense, it can cost us time, it can cost us money, and it can cost us our sense of well-being. About 1.5 billion per year in Europe alone is spent on health-related costs associated with allergy. So if we can mitigate this at all, that can be a great help to society. It could be doing so by having a very accurate forecast so people aren't exposed to pollen. Or if they have to be exposed to pollen, so they can take medication to not be affected as much by the high concentrations. So now that we know why we want to sample it, how do we sample it? Well, the most basic way is to use a Hurst-type trap. It's basically just a suction pump that sucks in ambient air at around 10 liters per minute onto a clockwork mechanism which rotates at around 2 millimeters per hour. Okay, this has a sticky substrate on it which causes the particles to adhere, and we bring this sticky substrate back to the lab and we use light microscopy to determine the species of pollen that are there and its concentrations. Of course, this takes a long time to do. It's very laborious. You need to be trained to be able to identify the different types of pollen, and there's several different types. As we can see here, there's a number of different morphological features and sizes of pollen. Okay, normally we're interested in poaceae or grasses. Okay, that's the one we traditionally link towards hay fever, but we should also be thinking about things like alnus and betula, which are alder and birch pollen, because 25% of people within Ireland who suffer from hay fever suffer from allergies caused by these type of particles. Of course, now, where do we sample it? Well, luckily enough, we've set up a few sampling sites around Ireland. We have one in Dublin, Carlow, Cork, and now Galway. Unfortunately, Galway and Cork have been shut down a little bit due to the COVID crisis, and the buildings that they were associated with had to close. But the sites in Dublin and Cork and Carlow are still running. And once you do that, you can get data like we see here. Okay, I just have plotted the pollen concentrations, total concentrations for 2017, 18, and 19 for Dublin. Dublin has been the longest established site. And we can see here that we have a whole host of different pollen types. We can see that urticaceae and poaceae over here on the pie chart on the right-hand side are the most significant contributors to total pollen. Okay, they're about 18 and 17% alike. Okay, and that's nettle and grass pollen. And we also have a significant amount of fraxinus, which is ash. We get a very different view when we look at Carlow. We can see that 80% of the pollen sample there was grass pollen. This actually makes a little bit of sense when we think more about the sites themselves. Carlow, housed in the middle of a field, okay, lots of grass around it, lots of meadows, uncut grass, so plenty of grass pollen. While the Dublin trap, situated on the top of a building, in a city which has a lot more ornamental trees associated with it. Of course, another part of this project, this POMMEL project, which looks at monitoring modeling, is we were able to get a few of unpublished data sets for pollen concentrations within Dublin. And we can see here we have concentrations from 1978 to 1980. And the total pollen concentrations, this top left-hand corner here, doesn't really change a whole lot. Okay, they remain fairly consistent. But if we actually look at individual species, they can be quite different from year to year. On the right-hand side here, we can see that there's quite a difference in concentration for poaceae, that grass pollen again, between 1979 and 1980, for instance, far higher in 1980. And of course, these are generally linked to meteorological parameters. Okay, so meteorological parameters will generally dictate how much pollen is released. And just to highlight this even further, okay, we've betula concentrations here for 3 different periods in Ireland, okay, at the Dublin site, 1978 to 1980, 2010, 2011, and 2018 to 2019. We can see the highest concentrations are in 2018, 2019. And the reasons for this, well, we haven't really answered that yet. Could be due to different, increased amount of sources near our site, but it also could be linked to climate. Okay, there's been an increase in carbon dioxide and increased temperatures. This could be the reasons why we're seeing higher concentrations of birch pollen now than we did back in the 70s. Okay, collecting this data then allows us to create a calendar, or a pollen calendar for Dublin and for Ireland, because different pollens are released at different times of the year. In this, there's a lot of data in this slide here, so I won't go through it all, but the red just indicates that they're highly allergenic types of pollen, the green, not so much. And then we can see at the different times of the year where pollen has been released. For instance, you can see here in the top left-hand corner, alder and hazel are generally released at the beginning of the year, January to March. While if we go all the way down to the grasses, kind of in the middle there, we can see, as you might expect for grass pollen, it's generally at its highest concentrations in June and July, okay? That's, hay fever, and, that's why it was initially called hay fever. Of course, we can also look at the phenological aspects of the release of these particles as well, looking at when the catkins are formed and when they're releasing pollen, or when the anthers are releasing grass pollen. Okay, I won't speak too much about this slide, but we can actually use that phenological analysis to determine a window of when we might see these pollen species. And we can see that in this slide here. That yellow trace in both graphs just indicates the phenological window when we should potentially see the pollens of those species. And then we can see here in the green trace, we can see the birch pollen species sampled by our sampler. One of the interesting things to note is that, well, the highest concentrations occur within our phenological window, but we also do get these significant concentrations before this phenological window. There's a couple of reasons potentially for that. Maybe our phenological window isn't perfect, we need to do more work there. I definitely agree with that. But it also could be linked to medium to long range transport of pollen. And this has been known to happen, has been shown to happen. And this got us thinking about looking at our data in another way. And we started looking at, analysing the atmospheric transport of potentially ash and birch around Dublin. Okay, we did this by creating footprints of the probability of where the particle originated from using high-spit 24-hour cluster back trajectories for very high concentration days of pollen, okay, above 80 grains per meter cubed during the main pollen season, when we would expect highest concentrations of these two types of pollen. And we get quite contrasting pictures, really. On the right-hand side here, we can see the probability of where the birch pollen was originating from. And we can see that, those red colours tend to situate themselves over Ireland. This kind of indicates that the pollen that we sampled on those days was probably from local sources. We get a different picture if we go to the left-hand picture there and see that that footprint actually kind of tails over the UK, indicating that some of those fraxinus or ash pollen might have originated from the Midlands of the UK. There's plenty of ash trees there, okay, collected by samplers over in the UK and potentially transported over to Ireland. So some interesting results there. Of course, another thing that this project tried to do was to map the sources of allergenic trees within Ireland. And we did this by combining several private forest databases, public forest databases, the Carling land cover from 2012, and a few other sources. And we were able to produce, the density of certain trees within Ireland, okay, indicated by the grayscale here down on the right-hand corner. Okay, very grey indicating 0%, while darker grey up to 75 to 100%. We can see for 8 different types of tree here, allergenic trees, we have mapped sources which could be very useful for dispersion modelling into the future. Of course, one thing I really want to talk about is, moving away from this Hurst volumetric sampler to maybe using more enhanced real-time methods of pollen detection. And the 2 instruments I want to talk about is the KH3001, also known as the Japanese pollen counter, and that's probably what I'll discuss it as, and then the WIBS Neo, okay, which uses, and they both use optical scatter, and in the WIBS Neo's case, fluorescence to determine whether a particle is biological or not. One of the main points about these two instruments is that they can do this instantaneously in real time. And when you do it for the Japanese pollen counter, okay, you can get, this is just an example of, the concentration data we got against time in the top left-hand corner. Okay, and you can see we see peaks in pollen concentration with time. And then if we get a diurnal plot of that, okay, between 12 and 12 for an average day during the campaign, we can see that, pollen concentrations tended to be higher during the middle of the day, which gave us, a bit of confidence in what the instrument was doing, because that's when you would expect pollen concentrations to be viewed by the instrument. Okay, and this has been used in the literature before, okay, particularly a few campaigns over in Switzerland. And what they did in those campaigns is they used different extraction windows, because essentially the Japanese pollen counter uses side and forward scatter to determine whether the particle is a pollen or not, then to determine the concentrations. And that's what we did. We used the extraction windows determined during those campaigns. So we were able to extract our potential pollen concentrations and then compare them with our traditional Hurst type method. And when you do that, you get kind of some nice results like you see here. Okay, in the top left-hand corner again, we see our Hurst concentrations of total pollen versus our Japanese pollen counter in the red trace. Okay, and they follow each other quite well, an OR of 0.51. We kind of tweaked, tried to play around with that extraction window as much as we could to see if we could make it any better, using what was seen in the literature previously again. However, it didn't really get any better. We got the only other one that was any close was about 0.47. And then we tried to use, just to identify single pollen species like Poese. And while we got an OR of 0.54, you can see quite obviously in the right-hand graph that there's a very high peak determined as a grass peak just before May the 15th. Unfortunately, that wasn't a grass pollen concentration, but it actually was a pollen concentration. So the instrument is quite good for total pollen, but not so good for speciating different pollen species. Moving on to the WIBS Neo, I just kind of want to give a bit more of an overview of how this instrument works, because it's a lot more dedicated towards bioaerosols than just using forward and side scatter like the Japanese pollen counter. Essentially what happens is your ambient air gets sucked in, gets hit by a diode laser at 635 nanometers. Okay, this gives us size and shape information, again using side and forward scatter and using Mie theory to determine the size of the particle. It's then hit by a xenon flash lamp at 280 nanometers, and its fluorescence is interrogated between 300 and 410, which is the FL1 channel, and 420 and 650, which is the FL2 channel. Okay, and these, if it fluoresces in those areas, they're named as FL1 or A-type particles, or FL2 or B-type particles. Then a second xenon flash lamp at 370 nanometers shoots the particle, and we get fluorescence between 420 and 650 again, and these particles are known as FL3 or C-type particles. Of course, we can then the particle exit the instrument, and the whole thing repeats itself, and it can do that up to 200 times per second at millisecond resolution, so far faster than the traditional techniques. Of course, you can then combine all this information on the particle, and if it's only fluorescent in the FL1, it's deemed an A particle. If it's only fluorescent in the FL2 particle, in the FL2 channel, it's deemed a B particle. But of course, it could be fluorescent in both. And this Venn diagram just shows you the 7 different characterizations you can have for fluorescent type particles. And that's the category analysis that we applied to our data. Okay, we had over 6 million particles for the campaign that we ran with the WIBS Neo, and we can see the preponderance of them are kind of B-type particles, as you can see here in the pie chart. Of course, you can also look at how that proportionality changes with time. We can see on the right-hand side here, we can see that it goes up and down depending on the day and the influences we have on our instrument. And of course, actually, most of the particles were actually non-fluorescent. Okay, so that's one of the nice things about the WIBS Neo instrument. It can give you, it's technically a particle counter as well. Only 18% of the particles, total particles counted, were fluorescent, while the remainder were non-fluorescent. Of course, one of the things we then did was to look at the size distributions of the particles for each of the fluorescent types. The WIBS Neo is able to look at particles between 0.5 microns all the way up to around 30 microns. As you can see here, we've just shown the size distributions for each of the fluorescent particle types. And you can see they're dominated by the smaller particles. And you'd expect this for any ambient sampling, especially when you're looking at such small size ranges of aerosol particles. something of interesting note is how different the ABC type particles are compared to the other type of particles. And ABC particles are generally linked with bioaerosols. Of course, we can also look at the size versus this shape factor. Okay, there's a lot of information on this slide here, so I won't spend too long on it. Okay, on the y-axis, we have size. On the x-axis, we have shape. 0 indicating a perfect sphere, 100 a perfect rod. Okay, and then the color indicating the concentration of particles. And again, for instance, the B-type particle right in the middle of your screen there, you can see that most of those particles are very, very small and very, very spherical. So they could potentially be, diesel droplets, for instance, fluorescing in the B channel, very, very small, particularly spherical. Well, if you look then at ABC, that's quite different. Of course, we try to compare the pollen concentration we got from our traditional sampler versus the WIBS Neo, and we saw that they didn't follow each other very, very well. Okay, a Pearson's correlation of about 0.5, so not too bad, but not great. We also tried to see if these WIBS Neo particles could be used for individual pollen species, and again, they didn't follow each other very, very well. Only a Pearson's correlation of about 0.5. I do have to note, however, this is towards the end of the pollen season. Okay, so the concentrations of pollen weren't very, very high, and Poaceae or grass pollen tends to be quite large, so the potential for wall loss within the WIBS Neo is quite large. This could affect it. The WIBS has been seen to correlate with pollen far better at higher concentrations. One interesting thing that we did note from this campaign, however, is that the WIBS Neo signal was very good versus fungal spores or total fungal spores. These can also cause allergy. As you can see here, the traces follow each other quite well. Pearson correlation at about 0.7. If we actually remove some of those fungal spores, if we remove Claudosporium, because it has been noted in literature previously that the WIBS has difficulties sampling that particular fungal spore, that even gets even better. The Pearson goes up to 0.8. And one of the main things I want to talk about within this POML project is our work on the development and refinement of pollen modeling. We use several different models and different methods to do so. For instance, we used artificial neural networks regression, support vector machine learning, multivariate regression, random forest, and a few classification models as well. And of course, we put several different pollen inputs within these. Okay, we use a concentration the day before, pollen concentration 5 days before, and even 10 days before. We also looked at the slope of pollen concentrations during the week before our forecast. I also previously mentioned phenological inputs, okay, and we showed the phenological window in a previous slide, but we also use things like growing days, okay, using a base temperature of greater than 7 degrees Celsius. And of course, we used a whole host of different weather parameters, okay, and I've shown a few here, wind direction, speed, cloud cover, maximum, minimum temperature, sunshine duration. We use a whole host of them. Combining all of these inputs into our models, we were able to see which model performed best. And we can see that on the left-hand graph in the left-hand top corner, as we can see here. In the black trace, we actually have our observed concentrations of pollen. And then in our other traces, we just have our different versions of the models that we created. Our artificial neural networks, our multivariate regression, our random forest, our machine vectors, and things like that. And we can see that at different times, different models do better than others. But what we found, okay, is if we combined all the models together using some form of ensemble approach, we got far closer to our observed values than if we did just using one model. And we can see that looking on the right-hand traces, we can see here. We have our observed concentrations for birch pollen in this case, and we can see that our mean, our median, and our weighted mean of our combined models actually are far closer to the observed than any individual model that we saw on the left-hand side. I mean, we can look at some of the statistics with regards to that. We have our Spearman coefficient here for our median is about 0.73, so it's the least accurate of all of them. Our mean model was about 0.77, and our weighted mean was seen to be 0.84. Okay, And this allowed us to get the most accurate reading. So kind of an ensemble approach is potentially the direction we'll go into the future. Of course, this is just for birch pollen. We need to do this for other types of pollen and for total pollen also. So now I'd like to just thank you. If you are interested in anything I've said today, you can contact us at Irish Pollen. We also have a website, which is www.pommel.ie, and we also have a project on ResearchGate known as Pollen Monitoring and Modelling, the Pommel Project. Thank you very much.